Hello students, let today revise about the process of cleavage formation and formation of blastula. After syngamy, zygote is formed and then the zygote starts to divide. Cleavage, it is a process of early mitotic division of the zygote into a hollow multicellular blastula. It does not involve the growth of the daughter cells. The cells formed by cleavage are called blastomere. Since there is no growth phase between the cleavages, the size of blastomere will be reduced with every successive cleavage. As the size reduces, the metabolic rate increases. Subsequent cleavages are thus faster than earlier one. This requires rapid replication of DNA and high consumption of oxygen. So let's see the process of cleavage. In human, cleavage is holoblastic. That is, since it has low yolk, the whole zygote gets divided. The cleavage planes may be longitudinal or meridional and equatorial or horizontal. Let's see longitudinal or meridional. This is the animal pole. This is vegetal pole. So here cleavage furrows runs along the medium axis, that is from animal pole to the vegetal pole, dividing the zygote into two cells. So this is longitudinal or meridional. It can be equatorial or horizontal, means the cleavage furrows divide the egg at its equator, running perpendicular to the main animal vegetal axis. It is radial and indeterminate. Let's see what is radial. It is a pattern of embryonic development in which the cell divides symmetrically along the planes that are either parallel or perpendicular to the central axis. This arrangement results in tiers of cells or blastomere that are stacked directly on top of one another. So either they are above or below. Indeterminate cleavage. In this process, the cells known as blastomere get separated from each other. Each one has potential to develop into a complete independent organism, that is indeterminate. The first cleavage in the zygote is meridional and occurs at about 30 hours after fertilization. It divides longitudinally into two blastomere. One is slightly larger than the other. The one blastomere is larger than the other. The second cleavage is also longitudinal but at right angle to the first one and occurs after 30 hours of first cleavage, then 60 hours after fertilization. The third cleavage is horizontal. After third cleavage, the embryo is in 8-cell stage. As the cleavages are going on, the young embryo is gradually being pushed towards the uterus. By the end of 4th day after fertilization, the embryo is a solid ball of 16 to 32 cells and externally looking like mulberry. This stage is thus called morula. The morula shows cells of two types: smaller clear cells towards the outer side and inner cell mass of larger cells. Cells are compactly arranged. Till the formation of morula, the zona pellucida is retained around the embryo and thus there is no change in the overall size from zygote to morula. The morula reaches the isthmus and gains entry into the uterus by the end of day 4.